Hello, it's Dr. Debbie Morris, and we are going to have a short talk today on part 3 of the endocrine system, and we're going to focus on the pancreas. In addition to this short lecture, there are going to be some case exercises with an assignment followed by a video with the explanations. So we'll do that after we do this lecture. Today's objectives are to describe what the products of the endocrine pancreas are, because remember that the pancreas has both exocrine and endocrine properties, and it'll be important to know the structure and the function of the key hormones that are made by the pancreas. We're going to talk a little bit about disorders of the pancreas and their consequences, and then, as I said, we're going to apply the knowledge of the whole module, endocrinology module, to some cases. The pancreas, while it is certainly an endocrine organ, is also part of the gastrointestinal system, and it has exocrine functions that are very important in the function of the gastrointestinal system. The exocrine products of the pancreas are enzymes that assist in digestion and are released through the pancreatic duct. So there are systems in the pancreas that are ducts that join with the common bile duct, and then the bile and the pancreatic enzymes together are discharged into the duodenum at the sphincter of Oddi. But the endocrine products of the pancreas are produced within cells that are called pancreatic islet cells. So the endocrine functional unit of the pancreas is an islet, or an islet of Langerhans. And here we have illustrated a pancreas. It has a duct system. The duct system joins with the common bile duct that is coming from the liver and the gallbladder. The products of the bile duct and the pancreatic duct join together and are released at the sphincter of Oddi into the duodenum. You can see that the placement of the pancreas is quite posterior. It is largely posterior to the stomach. It is actually a retroperitoneal organ like the kidneys. And here we are showing a small portion of the pancreas that is an islet of Langerhans. It has several different kinds of cells. I think that's all that we need. Okay. So this is another view of an islet of Langerhans within the pancreas. So there are cells around the islet of Langerhans that are making enzymes that are being discharged into these ducts, but the islet of Langerhans is surrounded by capillaries and has essentially 4 kinds of cells. Alpha cells, shown here in dark blue, beta cells shown in light blue, delta cells or D cells, and F cells that are also called D1 or gamma cells. So the alpha cells make an enzyme called glucagon. The beta cells make, did I say enzyme? Let's go back. Here we have a larger view of an islet of Langerhans within the pancreatic tissue. So it's surrounded by cells that are making enzymes and that are discharging those enzymes into ducts. But the islet itself is surrounded by capillaries. There are 4 kinds of cells in an islet of Langerhans: alpha, beta, or AB, D, delta, or F, which is also called D1 or gamma. And each of them produces a different pancreatic hormone, all of these hormones being polypeptides. The alpha cells shown in dark blue make glucagon. The beta cells make insulin, and you can see that there are more beta cells than any other kind. The delta cells produce a hormone called somatostatin, and the F cells make something called pancreatic polypeptide. While insulin is only made within beta cells in the islets of Langerhans in the pancreas, the other hormones produced by the pancreas have other sources as well. We're going to focus on insulin and glucagon, which are very important in the body's management of carbohydrate and are also important in protein and lipid metabolism. We talked about chemical messengers, and I mentioned that there were messengers that were hormone like substances but acted on nearby cells and functioned as something called paracrines. And the alpha and beta and delta cells in the islet of Langerhans that make glucagon, insulin, and somatostatin each affect the surrounding cells. So the insulin, glucagon, and somatostatin have effects on the production of the other hormones by the other types of cells surrounding them. The one that we're going to focus on the most is, of course, insulin. Insulin, as I said, is a polypeptide. It is made in the pancreas as a substance called preproinsulin. The preproinsulin has a peptide that is removed. That peptide is called the C chain. And then you have proinsulin, and the proinsulin joins by sulfite chains or sulfite bond by sulfide bonds into this polypeptide containing an A chain and a B chain. The C chain is released into the circulation. So this is all happening in the pancreas. They are released into the bloodstream as needed in a pulsatile fashion. There are a number of things that are involved in the control of insulin secretion. Some of them stimulate and some of them inhibit, and most of them are related to glucose metabolism and glucose levels. So the first and perhaps most obvious stimulatory controlling mechanism is when there is high blood glucose, the beta islet cells release insulin. And when there is low blood glucose, the beta islet cells don't release insulin. But there are some amino acids that trigger insulin release. There are some intestinal hormones, and we'll talk about those perhaps a little bit more as we get into the gastrointestinal segment. Acetylcholine, as a product of the parasympathetic arm of the autonomic nervous system, can trigger insulin secretion. And there are other things that inhibit it. I mentioned that somatostatin functions locally as a paracrine produced in the delta cells or D cells. And in addition, sympathetic output, so norepinephrine and epinephrine, inhibit the release of insulin. And there are a number of other factors that help to control insulin secretion. What does insulin do? Well, a big part of what insulin does is that it triggers the production of transporters that allow glucose to be transported through certain cell membranes, and particularly the cell membranes of muscle cells where glucose is needed for energy production, and adipose cells where it is converted into lipid for storage. In addition, insulin stimulates the enzymes that convert glucose, when glucose is high and insulin is being produced, into glycogen. Glycogen is a way the body stores glucose. It's essentially a polymer. It's similar to starch. It is made in the liver and in muscle cells, and when there is excess glucose, it can be stored as glycogen. When glucose is low, there are different enzymes that result in glycogenolysis, or the breakdown of glycogen. Insulin slows down gluconeogenesis, which we've mentioned previously, which is the production of new glucose from certain amino acids and fatty acids. It helps to regulate the production of lipids, lipogenesis, and it has a general effect to promote protein synthesis and growth. So insulin is an anabolic hormone. We said cortisol was a catabolic hormone and triggers the breakdown of proteins and of muscles. Well, insulin promotes protein synthesis and growth. This is an illustration of a beta cell, beta islet cell, where insulin is being produced and it's being stored in storage granules. And under the influence of assorted potential control mechanisms, the insulin is released into the extracellular fluid and hence into the circulation. And those can include chemical messengers and electrical signals. Here we are showing the pancreatic beta islet cells under influence of various controlling mechanisms. So increased plasma glucose causes production and release of insulin. Increased plasma amino acids, certain amino acids, also trigger release of insulin. There is a hormone called gastric inhibitory polypeptide that causes the release of insulin when food hits the stomach. We talked about how the parasympathetic neurons trigger the release of insulin. So parasympathetic signals acetylcholine, and how sympathetic signals, both from sympathetic neurons and circulating epinephrine, inhibit the release of insulin. There are insulin receptors everywhere, and they are a transmembrane receptor. Obviously, there are more of them on muscle cells and on lipid cells. When insulin attaches to an insulin receptor, it changes the shape of the receptor so that the part of the receptor that is on the intracellular side is able to activate enzymes in the cell called tyrosine kinases. And that tends to trigger ATP production along with other things like protein manufacturing. There are insulin receptors on the nucleus, on the endoplasmic reticulum, and the Golgi apparatus as well. One of the things that happens when insulin attaches to the nucleus and the protein making machinery of the cell is that a kind of receptor called GLUT4 receptors that are glucose channels are produced and mobilized to the cell membrane. And so we've talked about other ways in which glucose can be transported. For instance, there's a glucose sodium cotransporter, but GLUT4 proteins are actually glucose channels that bind glucose, change conformation, and release glucose into the cell. So here we have inactive insulin receptors. Here we have an insulin receptor, again a transmembrane receptor that has bound insulin and made the changes that allow signal transduction by tyrosine kinases and so forth, increasing the expression of certain genes that are going to result in the production of GLUT4 channels, and in addition, allowing the utilization of glucose and the synthesis of things like glycogen in certain places, the liver and the muscle, lipids, and proteins. So when insulin is released, a number of things happen. Blood glucose goes down. Why? Because one of the things that it triggers is the uptake of insulin into cells. Blood lipids and proteins also go down. There is an increase, particularly in glucose uptake by muscles and adipose tissue. We have an increase in the synthesis of glycogen in the liver and in muscle. We also have fat synthesis in adipose cells and protein synthesis in muscle. And all of these are helping to store excess energy for when it's needed. The opposite effects are inhibited. So lipolysis, or the breakdown of fat, is inhibited, gluconeogenesis is inhibited, and glycogenolysis is inhibited. We quit breaking down fat, making new glucose, and breaking down glycogen. Insulin increases the uptake of potassium by cells, and it increases the activity. It does that by increasing the activity of the sodium potassium ATPase. So here we show the things that insulin stimulates and the things that insulin inhibits. Stimulates glucose uptake, the breakdown of sugar, the synthesis of glycogen, the synthesis of protein, and uptake of ions like potassium. It inhibits the production of new glucose, the breakdown of glycogen, the breakdown of fat, the production of ketones, which happens when we're using fatty acids, and the breakdown of proteins. Let's talk about some of the pathologies that involve insulin. The one that most of you are familiar with is diabetes mellitus, and there are two types of diabetes. There's insulin dependent diabetes mellitus, and we also call that type 1, and there's non-insulin dependent diabetes mellitus, which we also call type 2. These days we're tending to call them insulin dependent and non-insulin dependent, but this is terminology that changes from time to time, so it's good to be familiar with both terms. In type 1 insulin dependent diabetes, the pancreatic islet cells are destroyed and they quit making insulin. Most often, this is a result of an autoimmune disorder in which antibodies to pancreatic beta islet cells cause destruction of the beta islet cells, and people with insulin dependent diabetes mellitus have low insulin levels, and the treatment is insulin. It's quite different from non-insulin dependent diabetes. Insulin can, under normal circumstances, bind to receptors and trigger the effects that it has. But in non-insulin dependent diabetes, there's insulin resistance in certain target tissues particularly, so that the insulin does not have its appropriate effect. People with non-insulin dependent diabetes can have normal levels of insulin, they can have high levels of insulin, and in some cases, often late in the disease, they have low levels of insulin. Most often, Non-insulin dependent diabetes is treated with drugs that either stimulate insulin release or upregulate insulin receptors. On the other hand, it can be treated with insulin as well. So even non-insulin dependent diabetes mellitus sometimes requires insulin for treatment. Extremely important to know that one of the effective treatments for most people with non-insulin dependent diabetes is weight loss. By reducing excess weight, we can reduce insulin resistance. The last of these pathologies that I'm going to talk about is an insulinoma. Sometimes there are small glandular tumors, adenomas, in or around the pancreas that secrete excess insulin not under the normal controlling mechanisms, resulting in low blood sugar. This chart shows only some, of the oral medications for diabetes. And the main reason that I have this here is not that you pay attention to them or learn them, but notice one, that they all are for only type 2 diabetes. There are no oral medications for type 1 diabetes. They have somewhat different effects or mechanisms of action. Sitagliptin, Januvia, increases the level of hormones to trigger insulin release from the pancreas, while metformin increases liver sensitivity to insulin. So it kind of helps fight that insulin resistance, particularly in the liver. And there are a few drugs that do other things. Precose slows the absorption of carbohydrates from the intestines. So it works not on the pancreas and not on insulin at all, but by slowing the absorption of glucose, which has certain obvious side effects. I want to talk about glucagon. Glucagon is the hormone that is made by alpha cells in the pancreatic islet, and it is in many ways the opposite of insulin. If insulin is the hormone that is produced when we are in a fed state, glucagon is a hormone that is sometimes called the hormone of starvation. Its release is triggered by low glucose, by a hormone called cholecystokinin, by fasting, and by extreme exercise, and it's inhibited by high glucose. When glucagon is released, blood glucose levels go up. In an opposite effect of insulin, glucagon stimulates glycogenolysis, the breakdown of that storage carbohydrate, and increases gluconeogenesis, or the production of new glucose molecules from lipids and amino acids. It increases the breakdown of fat and inhibits the synthesis of fatty acids from glucose, and all of these are essentially the opposite from insulin's effect. So here we're showing a balance with the pancreas on both sides. When blood glucose levels go up, there is an imbalance towards the production of insulin, which stimulates glucose uptake by cells, which stimulates glycogen formation. And all of that brings the blood glucose back to normal. When blood glucose levels go down from starvation, the pancreas releases glucagon, which stimulates the breakdown of glycogen and the production, gluconeogenesis, the production of new glucose, which brings the glucose back to the normal range. So in a fed state, insulin dominates, and in a fasted state, glucagon dominates. When insulin dominates, we use glucose, we make glycogen, we make fat, we make protein. When we're in a fasted state, when glucagon is the dominant hormone, we break down glycogen, we make glucose, and we make ketones because we are also using fatty acids for energy in the absence of glucose. We have to have gluconeogenesis, make glucose from fatty acids. I'm going to just briefly talk about somatostatin, which is synthesized in the delta cells of the pancreatic islet. Anything we eat, anything we ingest, triggers the release of somatostatin. But when insulin is being released locally by beta cells, it functions as a paracrine and inhibits the release of somatostatin. And somatostatin inhibits the secretion of both insulin and glucagon.